Let's now, in this chapter, review some of the details about specific cytological issues associated with the nervous system. Let's now look at a chart that summarizes the basic functional components of a neuron and the most common glial cells that are found inside the central nervous system. Typical neuron is a eukaryotic cell containing a common range of subcellular organelles that are features of any protein-producing cell. But neurons are unique in that they have long cytoplasmic processes. Dendrites provide the receptive area for the appropriate neuron. The axon, probably the most clinically significant part of a neuron, its job is to transmit action potentials, electrical impulses down its length so that they can reach the axon terminal and upon their arrival cause synaptic vesicles that are storing chemical neurotransmitter to release that neurotransmitter into a synapse and cause some sort of postganglionic effect, either with reacting with another neuron or some kind of target tissue. The non-neuronal cell types in the CNS, we've discussed earlier, include oligodendrocytes that form myelin for any axon inside the neural tube derivative components, including the optic nerve. Astrocytes, they are the scaffolding or the connective tissue in a way of the central nervous system. They control the microenvironment of neurons, and with their foot processes, they help maintain the blood-brain barrier, which is largely formed by tight junctions between capillary endothelial cells. Schwann cells are a peripheral nervous system myelinating cell. We know that they are derived from neural crest cells, and their ability to form myelin is much more restricted than that of a typical oligodendrocyte. Ependymal cells are ciliated neurons that are involved in the circulation and flow of cerebrospinal fluid because they are the lining epithelium of the ventricles and the canals, including the central canal and the cerebral aqueduct that interconnect the ventricles. Microglia are unique. They are not derived from ectoderm. They are derived from mesoderm. They are part of the mononuclear phagocyte system, and they act as central nervous system macrophages in a way. Now, let's look at a little more detail about the neuron itself. Here's a picture that illustrates a typical, in this case, motor neuron that has its cell body in the central nervous system and its axon in the peripheral nervous system. And as we indicated, a typical neuron has an array of eukaryotic organelles, including nucleus, nucleolus, Golgi, endoplasmic reticulum, lysosomes, and so on. But particularly since neurons are utilizing a neurotransmitter as a way of communicating with other neurons and target tissues, they're going to have prominent amounts of rough endoplasmic reticulum, which in neurons is known as Nissl substance. But note that Nissl substance, whose job is to produce proteins in the form of neurotransmitter that are bound for export from the cell, Nissl substance is only found in the cell bodies of neurons and in the proximal parts of their dendrites. What this means is, since Nissl substance is not found in axons, neurotransmitters and precursor of neurotransmitters must be transported down the length of the axon by a transport mechanism to allow the neurotransmitter then to be taken up and stored in synaptic vesicles until arrival of the appropriate action potential. So all axons of neurons utilize a phenomenon known as anterograde axonal transport, whose job fundamentally is to transport precursors, or actually the neurotransmitter itself, from the cell body down the length of the axon to the axon terminal. But as we'll see, all axons also may utilize a phenomenon of retrograde axonal transport. This is used mainly as a recycling mechanism where an axon terminal may take up substances in the synaptic cleft and transport that substance maybe back to the cell's lysosomal system or to recycle membrane, synaptic membrane, and the like. So axons utilize a bidirectional transport mechanism, anterograde going from the cell body to the axon terminal, retrograde going from the axon terminal back to the cell body. And before we look at the details of the transport mechanism, the other point that's certainly made on here again is that an axon, depending on where it is found, will be myelinated by either an oligodendrocyte if it's in the CNS or by a Schwann cell if it's in the peripheral nervous system. The additional feature that we need to make about this is that only axons in nerves that are myelinated or encompassed by Schwann cells have the capacity to regenerate if severed. So First, let's look at the transport mechanism and the subcellular organelles that mediate its process. As we said, axons utilize a bidirectional axonal transport in the form of anterograde going away from the cell body toward the axon terminal, or retrograde going from the axon terminal back toward the cell body. Note that the anterograde transport and retrograde transport both utilize microtubules, and both are energy-requiring processes in that anterograde axonal transport utilizes an ATPase motor molecule known as kinesin, whereas retrograde axonal transport utilizes dynein. And note that because mainly axonal transport in the anterograde direction is transporting neurotransmitter and its precursors, it occurs at a very fast rate, at the rate of about 400 millimeters per day. But note that there's also a bulk flow, mainly of cytoplasm of the axon, that occurs in the anterograde direction. That's much slower, doesn't require the energy source of kinesin, and bulk flow down the length of the axon occurs at the rate of only 1 to 3 millimeters per day. Retrograde axonal transport, certainly slower, used mainly in recycling and transport of substances back to lysosomes, occurs at the rate of only about 200 millimeters per day. But it is the retrograde axonal transport and the aberrant use of that is the mechanism by which neurons can be affected by the herpes and polio viruses, rabies, and tetanus toxins that affect either motor or sensory neurons as a result of aberrant retrograde axonal transport. Next, let's look at exactly what happens when you sever an axon, regardless of whether it occurs in the peripheral or the central nervous system. When there is a complete severing of an axon, two things happen. One is the part of the axon that is distal to the cut, regardless of whether it is in the peripheral or the central nervous system, undergoes what is known as anterograde or Wallerian degeneration, and based on that, completely degenerates. The axon that is proximal to the cut and still attached to the cell body itself undergoes retrograde degeneration. And as part of the retrograde process, the neuron cell body belonging to the cut axon will undergo what is known as chromatolysis, where typically there will be a swelling of the cell body itself, a dispersion and engorgement and elaboration of the Nissl substance as the cell body is attempting to gear up to regenerate its axon. So in the picture on the next slide, we see a later stage manifestation of the same process where we've cut the axon, we have seen it undergo anterograde degeneration and retrograde chromatolysis, and this makes the point that only axons that are myelinated or surrounded by a Schwann cell will provide a sheath down which a cut axon can regrow. So it's the presence of those Schwann cells that provide a kind of a tubular trough down which only a peripheral nerve axon can regenerate. And note the rate of degeneration. The rate of degeneration corresponds to the slow rate or bulk flow rate of anterograde axonal transport, because the regeneration rate only occurs at the rate of about 1 to 3 millimeters a day. Whereas the other point is, if you cut any axon that is myelinated by oligodendrocytes, the axon of that neuron does not have the capacity to regenerate. And this is the point that's made that summarizes in text form. Basically, regardless of where you cut an axon, doesn't matter whether it's cut in the central or the peripheral nervous system, that axon that is distal to the cut will undergo anterograde or Wallerian degeneration. And we make the additional point that the closer the destructive lesion is to the cell body, the more likely the neuron, particularly in the peripheral nervous system, is likely to die. But again, only if the axon was enveloped either by the myelin sheath formed by a Schwann cell or is enveloped by the endoneurial sheath formed by a Schwann cell in the peripheral nervous system, will that peripheral nervous system axon have the capacity to regenerate because of the scaffolding or the sheath-like tube that the Schwann cells provide to provide a tubular directionality to allow severed axons to regenerate. And again, we make the point that the rate of regeneration is very slow. It's proceeding only at the slow bulk flow rate of anterograde axonal transport, at basically at about 1 to 3 millimeters per day. And then finally, as a preview of what's to come, let's just look at a couple of views of the central nervous system. First, in a mid-sagittal section shown here, we're looking at the medial aspect of the brain. And certainly we can see that in a medial aspect, it's dominated by the telencephalic derivatives, the medial aspect of a corresponding hemisphere. What's shown here in kind of the lighter gray shaded area is the cut edge of the third ventricle. And situated out in the lateral wall of the third ventricle are the components of the diencephalon that include the thalamus, the largest part, hypothalamus below it, and obviously closely related to the hypothalamus will be the mammillary bodies and the posterior pituitary. Then extending below that third ventricle will be the cerebral aqueduct that courses through the midbrain. You can see the fourth ventricle separating the overlying cerebellum from the pons of the medulla, and then the spinal cord, even though it's not labeled, continuing down into the vertebral canal. And again, we've noted some clinical significance with regard to some of these. Some of these we've talked about, some of them we'll discuss subsequently. Note that the exam questions in many versions will show an MRI of the medial aspect of the central nervous system, like this one, and describe clinical scenarios and ask you where the problem is or where the deficit is or where the degeneration is. And some of the common ones that we'll talk about subsequently will be different forms of pituitary tumors, craniopharyngiomas and adenomas, and how they tend to compress the optic chiasm, which you see situated directly above the pituitary. We'll talk about the degeneration that typically occurs in thalamic and hypothalamic neurons, including the mammillary bodies, in patients that have thiamine deficiency and Korsakoff syndrome. And when we deal with parts of the brainstem, we'll talk about Parinaud syndrome, which is the result of a pineal tumor that compresses the area around the superior colliculus in the dorsal part of the midbrain. And then finally, an inferior view of the brain. It provides a very good illustration of the ventral aspect of mainly the brainstem and the cerebellum. And as we'll see when we review the brainstem, that the brainstem is home of 9 of the 12 cranial nerves that enter or exit in very specific patterns, even though it seems like a random array. They enter or exit in very specific patterns relative to various parts of the midbrain, the pons or medulla.